Welcome everybody back to the first Breeding Insight OnRamp seminar of 2024. So welcome back to this seminar series. Again, the name of that is Opportunities for Integrating New Tools into the Breeding Pipeline. And it's really great to see everybody after winter break. And we have a couple of talks scheduled already. And I'm really looking forward to hearing what our presenters have to share. So today I'm excited to introduce Dr. Lorenz. He's a professor at the University of Minnesota in soybean breeding and genomics, and works closely with the Northern Uniform Soybean Tests in cooperation with the USDA. He will be sharing some of the resources his group has developed to advance genomics and genomic selection in public soybean breeding. And kind of as we've done things in prior seminars, if you could please wait and hold questions until the end of the presentation, just to keep in mind our time limits. And you can post questions in the chat throughout the seminar if that's easier. And we'll take both questions and read off questions in the chat at the end. So with that, I'm going to hand it over to Dr. Lorenz.
Okay, great. Thanks a lot, Heather. Thanks to Amanda, Heather, and Keo for the opportunity to give this presentation today. And I'd like to first acknowledge my co-authors. Vishnu Ramasubramanian is a postdoc currently in my group who's contributed a lot to the development of this work. Clayton Wartha was a former student in my group who's also contributed some data to this work, as you'll see a little bit later on. Rex Nelson, the USDA at Soybase, has been very instrumental in helping us developing data resources and storing data and extracting data. And of course, Amanda, Heather, and Keo have been great partners in terms of adopting breed base to soybean in a new database that we call Soybean Base, which we've been using. And then Sushan, Ben, and Liana were former postdocs in my group that have contributed data and results to what I'll be presenting today. And then finally, I'd like to acknowledge all of the Northern Uniform Soybean Test associated public soybean breeders who have contributed data to this resource, and many of which I actively work on projects such as this. So. All right, let me just start off here by kind of giving a really high-level overview of what my University of Minnesota soybean breeding program attempts to accomplish. So at a very high level, our mission statement is basically to advance soybean cultivar development through research, education, and outreach. And we try to do that by combining an applied variety development program with research on the genetics of new and upcoming important traits, or maybe previously important traits on which we don't know the genetic basis of very well. So we do a lot of research into, I guess, just the inheritance of those traits and the genetic control of those traits. And then we try to combine that with research on breeding methodology, which I'll be largely focusing on today. And then, of course, we educate a lot of graduate students in this overall context who hopefully go out and largely do go out and become leaders in their field. So like I said, this is a broader context and in terms of our overall program, but today I'm specifically going to be focusing on a couple of projects related to breeding methodology development. And I also want to point out that this all is occurring in the context of an active variety development pipeline where we make crosses and perform single seed descent, and we test potential varieties or breeding lines throughout a number of years at multiple locations. And hopefully we get new varieties at the end. Usually we focus on developing new germplasm for parental stock for other organizations, as well as specialty type or food type soybean varieties. And so a typical breeding pipeline is just like ours, where again, we're developing populations through a combination of summer and winter nurseries. And then we have a number of stages of testing, starting off with looking at things at the progeny row phase and largely making visual selections and selections based on molecular markers. And then we have a couple of years of internal testing. We have a year of preliminary trials, our first year of yield testing, and then those go on to another stage of internal testing, which we call the advanced yield trials. And then, as I'll talk a little bit about later on, we enter the best lines from those internal trials into our USDA coordinated regional trials. And it's through these trials that we're able to achieve a much larger number of locations, so we can really sort of sift out those truly superior lines that have good and stable performance over a broader geographical area. And then, of course, we have to perform the task of seed multiplication and purification, and we have a nice testing network within Minnesota, and we have a good research and outreach station network that we can use and cooperate with. So that's just a little bit of context here in which all this work is occurring. So to get more specifically to the topic that I'm talking about today, and that is genomic prediction and selection. Hardly needs much of an introduction these days because it's been, a methodology, a technology that has been around for nearly 15 years. But nevertheless, just to frame this, genomic prediction is essentially the combination of genome-wide marker data combined with phenotypic data. And then you combine that data together to basically train a prediction model. And the important point here is that those prediction models do not take into account any sort of QTL mapping or marker selection. So QTL mapping, marker selection is not performed. We're not selecting QTL based on any sort of significance threshold. We're simply using all the genome-wide marker data together at one time in a lot of different types of models that can handle higher dimensional data than a more typical least squares type of model. So, and then once we get those predictions from that model, we will genotype our selection candidates, and we'll make predictions on those, and hopefully our predictions are good enough so that we can make selections on those predictions without maybe even seeing something in the field, or maybe combining our predictions with observations in the field to get a better estimate of the genotypic value or the breeding value. So in the context of a breeding, let me move this bar here. In the context of an applied variety development program, there are several phases or several stages at which you could apply prediction to increase the efficiency and the speed of your program. So, for example, at the top here, you could possibly use predictions to help better choose your potential parental matings for which you want to make crosses and derive new populations. You could perhaps actually select early generation plants here that haven't been inbred yet, and perhaps recycle those very quickly as parents if you have a good estimate to the breeding value, and really drive that genetic gain, speed up that genetic gain by reducing that time factor. Or you could reduce the number of yield trials you have to perform at the PYT and the AYT stage by genotyping and making genomic selections on progeny rows. A lot of times, especially for elite by elite crosses, it can be quite difficult to make a visual selection among a bunch of different progeny rows. And having a prediction perhaps would allow you to better make selections at this phase. A phase at which, since we just have one single row, the heritability of that yield phenotype would be quite low. So a prediction could be very useful at this phase to enrich your yield trials for really those truly higher yielding breeding lines. You could also perhaps leverage prediction to accomplish what's known as sparse testing. Perhaps rather than having replicated trials at multiple locations where everything is balanced, say, for example, a breeding line is tested in 2 replications at 2 locations, and you have a nice balanced trial, you could perhaps make that unbalanced, and so that you have perhaps a set of breeding lines at one location, a different, perhaps overlapping set of breeding lines at another location, maybe unreplicated with some sort of spatial or augmented type of design, and then leverage what we call the genomic relationship matrix calculated through the genome-wide markers to share information across all those breeding lines and get a prediction. And that could allow you to possibly evaluate more selection candidates or breeding lines under the same number of total area or number of plots you'd have to actually manage and harvest. There we go. So there's countless simulations out there suggesting that genomic selection holds a lot of advantages, especially when you can reduce that time factor. So a study published a number of years ago now looked at this breeding method that basically imposed rapid cycling genomic selection, where parents were, parents were basically single F1 plants, and they were genotyped and selected and used again as parents as quickly as possible. So they called that rapid cycling GS. And that was actually decoupled from the overall genomics, the overall variety development pipeline. So this rapid cycling GS basically enhanced the value of that germplasm as quickly as possible by making selections, making genomic predictions and selections as quickly as possible and recycling that germplasm as quickly as possible. And so these authors showed that over time, of course, since you've reduced time so much in terms of your selection cycle, you're able to achieve much, much greater genetic gain than you would with something like phenotypic selection, where selections are made further down in the pipeline and where it would take possibly multiple years to do that. And then in between, we have other types of genomic selection methods, such as making genomic selections at the PYT stage, or perhaps the plant row stage, or what they call the head row stage, and so forth. So basically time, right? Using genomic selection to reduce time before you have to choose a parent. The other application or use of genomic prediction could be to basically broaden your scope of selection. So, as a phenotypic plant breeder, you can only make so many crosses per year. You can only handle so many crosses. But among all the different breeding lines you have out there, you have an enormous number of possibilities that you could make, right? You can make perhaps tens or hundreds of thousands of possible crosses among all breeding lines available to you. You can only make, of course, a subset of those. But if you can predict the value of each one of those crosses by having a genotype of every single potential parent and having a training population on which to train that genomic prediction model, you can predict how all maybe tens of thousands or hundreds of thousands of possible crosses could perform, and you could select your crosses based on the prediction of those crosses. The same applies with germplasm resources, G by E, and hybrid performance, where you can, again, through the power of prediction, predict all possibilities in terms of how all possible varieties could produce at all possible environments, or perhaps in the case of a hybrid crop, how all inbred lines would come together to form a certain hybrid. So that kind of, in terms of breeding crosses, that kind of boils down to this genomic prediction methodology known as genomic mating, where now we're basically going to genotype all the potential parents in our crossing block, and we're going to predict the mean of the resulting population from that cross, the variance of the resulting population from that cross, and we can even go on to predict the genetic correlation between traits for all possible crosses among all the breeding lines we have at our disposal. And so the basic idea here, of course, is that when we think about a desirable breeding cross, we want a combination of a high mean of the population resulting from that cross, as well as a high parent, because the most important feature about the progeny from this cross is going to be basically the performance of the superior progeny. The performance of the superior progeny is represented out here on the tail of this distribution. So you can accomplish this either by having a high mean or having a high variance, and ideally you would have both. And so that tail ends up being out here someplace. And once again, you can go on to expand that to include not just the variance, but also if you're thinking about multiple traits, you can also predict perhaps the genetic correlation among traits. And so you can find combinations of parents that produce new breeding populations to perhaps maybe even have a more favorable genetic correlation between the traits that you're interested in. And so in this case here, this is a specific example where we have a training set developed from some historical data that I'll talk about in a minute. And then we have a bunch of potential parents in our crossing block. And the idea here is that we train a model and we predict the features of these resulting populations, such as the genetic correlation, the mean, and the genetic variance. And then among all these 14,000 and more possible crosses, we can then use this information to really select those few that we can actually handle, and those that are the best based on these predictions. And so we've done that in the past. And so you can see here we have plotted the superior progeny mean, the predicted superior progeny mean of our traits that we're interested. In this case, it was yield in soybean and protein in soybean. And what we want here is we want populations that are predicted to have a high superior progeny mean, which again is a function of the overall population mean and the genetic variance of that resulting population. And we want predicted populations that sort of exist in this part of this distribution. We want populations that are going to have a high superior progeny mean for protein, because we want high protein. We also want a high superior progeny mean for yield. And so it's these crosses here that are colored are the ones that we determined were the most suitable to make for this experiment up in this part of this distribution. And so you can see that, you know, as is well known, that protein and yield in soybean, as it is in other crops, is negatively correlated. So you want these up here that have the best combination of both of these traits for this particular objective. And we made those crosses in the field. Here are the parents that we chose, and we basically chose them on the basis of their superior progeny mean for yield and protein. And we derived populations from those. And a couple of years ago, we derived breeding lines from these populations. And this is just a picture to show you that they do exist and they actually look pretty good in the field. So that's on the basis of some work that we did a few years ago. I did a few years ago in collaboration with Jeff Nyhart, who was at the time a student with my colleague here at the University of Minnesota, the barley breeder Kevin Smith. And what Jeff asked was simply, you know, essentially that. Can you form a training population, and can you predict the genetic correlation of a cross? And can you choose potential crosses based on more favorable genetic correlations? And so you basically need the marker effects for this. You need the genetic distance between the markers and so forth, and plug into this equation here, which I don't need to go over. And I'm going to gloss over all these details. That was what we tried to do. And you can see within this population that Jeff worked with a number of years ago, there was a range in terms of the correlation between these traits that he looked at. One trait he looked at was one combination of traits he looked at was the severity of fusarium head blight resistance and heading date. You can see that among all these many thousands, tens of thousands of potential crosses, you can see that there is a range of predicted genetic correlation between these traits, ranging from near -1, which is highly unfavorable, and sometimes ranging above 0, which is relatively favorable. So you can see that these ones we wouldn't want, where there's a strong correlation between heading dates and FHB severity. But in some cases, we could find populations, predicted populations, that may have a more favorable correlation. So those would be the ones that we would like. So what Jeff did is he ran a series of simulations to see the accuracy of predicting these different properties of a distribution from a distribution representing a new breeding population. And you can see that under these different scenarios, in terms of different levels of heritability for the combination of the traits, trait 1 and trait 2, and under the scenario of a genetic correlation generated here by simulating loose linkage between loci controlling these traits, you can see that as expected, we can do pretty well in terms of predicting the population mean, in terms of predicting the population variance here. For trait 1 and trait 2, you can see that the correlations are, of course, less, but still not too bad. And even for, if we have a large enough training population size here, even for a genetic correlation, all right, which is going to be a function of the genetic variance, as well as the covariance between these traits, you can see that we have the overall predictive ability here is lower, but if we have a large training population, it's actually not too bad. So there is some information there, is what this suggests. So we went on to do some additional simulations and under certain different scenarios here of training population sizes and correlation architectures and genetic correlations and heritabilities of the trait. And so what Jeff did is he had a pool of selection candidates. He had potential parents, and he had potential crosses, and he selected these different crosses in the simulation based on a model that accounted for basically the correlation between the traits, or what he called the correlated progeny mean. And then he had cross selections that he made only on the basis of the family mean, so not taking account of the correlation between the traits, and then random selections, random cross selections after the parents actually had been selected. And then he repeated that process 10 times to look at the overall genetic gain accomplished through these different methods. And so what we found is that when you do account for the correlated progeny mean, and you look at the two traits, the response of the two traits in the form of an index value, you can see that when we account for the genetic correlation between the traits, so we actually get a better response over time compared to only making selections on the basis of family mean or only making random cross selections here. So there seems to be an advantage, and much of that advantage has to do with the second trait here, where the second trait, perhaps if there's a negative correlation between these traits that's being simulated, the second trait doesn't decay as much in performance as the first trait here is being improved. So we're getting the boast of we're better maintaining performance in the second trait. So that's all great, but as I said before, when we did this in real life, we didn't have any good validation. When we, in this case, in the barley example that Jeff worked on, this is a computer simulation, so there's some value there, but it's still be nice to evaluate this using real data. And it'd also be nice to look at this in soybean because I work on soybean. So what we did is it's hard to validate something like this because to really validate something like this, you need lots of different families. You need really big families because you need an accurate estimate of the genetic variance. You need an accurate estimate of the genetic correlation. You can't do that with just a few dozen individuals in a family, like a lot of our public programs represent. By the time something makes it to a yield trial, you may only have a few dozen, likely less than 50 individuals that represent any one cross. So we looked for existing data resources to help validate this, and the one that seemed like it was the most suitable, although there are some disadvantages here, was the existing soybean nested association mapping population here that had been developed through support of the United Soybean Board and the North Central Soybean Research Program, and was worked on led by Brian Deers, the University of Illinois, and Jim Specht at the University of Nebraska, among many others. And so this is what the soy NAM population looks like. It's a very typical NAM structure where you have one single hub parent and then a bunch of different founder lines. And from each of these 40 founder lines here, you derived, I think in this case, 140 different recombinant inbred lines for a total of 5,600 recombinant inbred lines. And these were evaluated at up to 14 different environments. And so we have good data. And so what we did is we basically did a leave one family out type cross validation where we trained a model using, we were able to get the genotypic effects for each of the reals using a proper statistical analysis. And then we're able to essentially use a leave one family out cross validation where we would train a model on the basis of all the information except for that one family we left off. And then we would predict the genetic correlation among these traits within that one family, as well as the genetic variance for all the traits and the mean of all the traits among all possible 40 here. And so these were what these distributions look like for each of the traits here. So you can see that among it's actually 39. There's one family that was lost. So we actually have 39 different populations here. So among these 39 different populations, you can see that we have different shapes in terms of which would be represented here as the variance and different means. And so what we want to do now is we want to see if we can use a genomic prediction model to predict the means and the variances and the correlations among the traits among all these different 40 families. And these are what these results look like. So in terms of predicting the mean here, this now would represent the correlation between the predicted mean and the mean estimated from the phenotypic data using that leave one family out cross validation I discussed. You can see that for mean, we have a significant correlation coefficient here in all cases. Sometimes the correlation coefficient is moderate to high. Sometimes it's a little bit lower than I would expect for a family mean. For the genetic variance, you can see that we're only successful in predicting that genetic variance among those 39 different families here in about half of the cases. Days maturity, we had really good success with. Plant type, pretty good success with. And seed size, pretty good success, and a lower but still significant correlation coefficient between predicted genetic variants and observed or estimated genetic variants on the basis of the phenotypic data for seed yield. These are those scatter plots right here. You can see that, for example, days to maturity has a very high correlation, but that's largely happening here because we have a lot of leveraged data points here, where we have kind of a cluster here that has both an observed and predicted performance, predicted low genetic variance, and then a cluster up here for which the observed and predicted genetic variance was high, and a couple points in the middle. But nevertheless, our model did tell us which ones were low and which ones were high quite successfully. Seed yield is down here. You can see a little more of a uniform, evenly distributed distribution of those 39 different families. Same for seed size and plant type. Again, we have a little bit of clustering going on here. But we could predict, for many of these traits, we could fairly accurately predict the genetic variants. At least there's information there. So in terms of the all possible pairwise correlations among all the different traits, we have, days maturity in lodging, days maturity in oil, and so forth and so on. Here in this table, you can see that of these 24 possibilities, 18 had significant correlation coefficients. That means we were able to, at the level of statistical significance, we're able to predict the genetic correlation between these traits with, again, these predictive abilities ranging from about 0.68 down to about, I guess in this case, only 0.27. So there's a range there, but they're significant. You can see that here's some different examples where these are the genetic correlations between oil and seed yield. We were able to predict this with a moderate amount of accuracy, same for plant height and seed yield. Actually, the correlation we're most interested in was protein and seed yield, and this one we were not able to successfully predict in this case. So we cannot predict very well the correlation between protein and seed yield among these different families. And that could be that there was not very much variation in protein within these different families for whatever reason. So moving on from that, in terms of opportunities and validation of using genomic mating to enhance the breeding program, we've also played around a little bit with performing genomic selection in our progeny rows. And so here's an experiment we did a couple of years ago where we genotyped 2,500 progeny rows in a breeding program, and we selected some at random. We selected some using genomic prediction, and we did this on the basis of a historical training population. And then we placed these in validation experiments the following years to validate this. And so just a little bit of background here in terms of our historical training population. The historical training population came from these uniform soybean tests here that have been performed by the soybean community and USDA over the last many years. And the objective of these tests here is to essentially, again, as I mentioned before, do this regional testing so we have a better idea of the potential for a new variety when released. And again, they come in these reports every single year that we as a soybean breeding community look at and examine, and we depend upon this information to have enough data to release new varieties. And so they occur, the northern uniform soybean tests here occur throughout the upper Midwest, and there is also a version of this in the South called the southern uniform soybean tests. And these are currently coordinated by Guhan Kai and Adam Brock in the north, and Ann Gillen coordinates those in the south. So we took all this historical data and we turned it into an electronic database and put that into a database to use as a data resource for projects such as this. And we've been genotyping these lines, entering these regional trials now for some time. And so we have genotype data and phenotype data in analyzable format. And so we can kind of look to see, okay, how good can we perform genomic predictions within these different tests? And you can see that if we take a leave one test out approach, and so we take one whole test, which would be a balanced set of lines tested at anywhere between 5 and 15 different locations. We set that aside We remove that from our data set, and then we train a genomic prediction model using the remaining data in the historical data set. You can see that our prediction accuracy is actually quite good most of the time. So we've been able to achieve prediction accuracies ranging all the way up to 0.95, which seems really quite high. But most of them are good, being well above 0.5 in the vast majority of cases here. So that suggests that we're doing pretty well on this using this historical data set. But how well does it do when actually applied to our real program? And so once again, bringing you back to this experiment, we were actually able to genotype progeny rows in our program, and then we performed genomic predictions using this historical data set. And these results haven't been fully analyzed yet. We're still adding data to these results, but a quick look at this would suggest that it kind of depends. And so these would be the results from the validation experiments performed at different locations within Minnesota. And what I did here is I took the best top 10 lines based on that validation data in the following year. All right, so if you look at the best, because really what we want to do is we want to use predictions to select the best lines. We can use predictions to select the best lines, then we can essentially drop out lots of other lines on the basis of predictions and not spend our resources testing those. And so what you can see now is we have these best top 10 lines, and you can see that in the north, our results were kind of mixed, where we kind of had an even distribution here of random and genomic prediction. As many random lines ended up in our top 10 as the lines selected on the basis of genomic prediction. Same went for Morris. In Rosemont, genomic prediction did better. We only had 2 lines from the random selection procedure sneak into our top 10. But what I was really encouraged about was the performance in southern Minnesota, both at Lamberton and Waseca, where actually we had the best data and where our data was best connected to the training population because of the later maturity of those breeding lines and the amount of data coming from southern Minnesota and from places like Iowa. I think we had the best predictions in the South. You can see that the top 10 lines in our validation trial here were almost exclusively selected by genomic prediction, and only 2 snuck into the top 10 across both locations on the basis of random selection. So again, better connection in the southern Minnesota to the training population. I think that has something to do with it, but we still need to further work with these data. In terms of future directions, some things I want to use genomic prediction for in my program would include better predicting genotype by environment interactions. And then that leads into a possibility of using genomic predictions to effectively increase my testing environments, even in the early stages of my yield testing program. So once again, I mentioned that in the PYT stage in our program, we typically only evaluate those at 2 locations because we want to test as many as 1,500 or more different breeding lines. And so we can only really use 2 locations, just the resource problem. When we go to our AYTs, we only have 3 locations. When we get to our regionals, we have many locations, but we only have a small number of lines. And so what I'd like to do is I'd like to leverage marker effects shared across different sets of germplasm. I'd like to leverage genomic relationships shared between advanced testing lines and preliminary testing lines to effectively increase my number of testing locations. And so if we can connect individuals here in our regional trials through the genomic relationship matrix to individuals being tested in our preliminary trials, perhaps we can better effectively predict the performance of these early generation lines in our PYTs in terms of how they would perform at all these testing locations being accomplished by the regional yield trials. And so these would be predicted performances, but because there would be relationships, there would be connections between my PYT lines and my regional yield trial lines, and these PYT lines would be tested at the same location as my regional yield trial lines. I have connection both in terms of shared environments and shared relationships, and I can maybe get a prediction on how each of these PYT lines would perform at each of these regional trial locations, even though they actually weren't tested there. And so if the predictions are pretty good, then we could effectively increase our testing locations, even in the early generations, and perhaps make much better selections than what we can make currently using only 2 locations. So that is a future direction I would like to move. The other direction I would like to move again is doing a better job of predicting G by E. And so the deal is that it's pretty easy to predict tested genotypes in environments that I've tested. And it's less easy to make predictions on tested genotypes in untested environments or maybe future environments. And likewise for tested environments and untested genotypes, I haven't actually seen a genotype in the field. It's less easy to predict that even in a tested environment. But what is really hard here is predicting untested genotypes in untested environments. So individuals in my program that haven't been in the field yet, perhaps they're at the progeny stage, perhaps they're just an F4 plant. Maybe I want to predict how they would do in future environments. And that's very difficult because I need to connect my tested environments to those untested environments using environmental covariables. And I think this is particularly important because what we really want to do in a breeding program is we want to predict how things do 5, 10, 15 years down the road. And this is especially important in the face of climate change, where the climate is rapidly changing. Summers perhaps are warmer, drier, wetter, whatever it might be. It'd be nice to use a climate change forecast model and leverage a G by E prediction model to predict how my perhaps F4 plants would perform in the environments that exist 10, 15 years from now. Maybe not that far, but 5, 10 years from now, or maybe even further for some far out predictions. All right, so that's all wonderful, but the question is, how do we make this easier for academic and public programs to accomplish? So some challenges that are unique to academic programs, I think, well, maybe not totally unique, but things I think about, and I think some of these things are kind of unique to public programs. One would still be cost of genotyping. We talk a lot about how genotyping is becoming cheaper and cheaper, but if I want to do genomic prediction on 10,000 progeny rows, the cost is still too much for that. It would cost me hundreds of thousands of dollars to do that, and we just don't have the budget for that kind of stuff. Turnaround time, so we get data and predictions quickly enough so we can make selection decisions in an applied program. Human resources is always a challenge because a lot of the folks who have the training and the interest in running genomic prediction models and doing all the genome-wide marker analysis and the quality control and the bioinformatics and so forth tend to be they're in academic programs. They tend to be students or postdocs who are temporary here by nature, and then they go on to more permanent jobs. And so they leave the program and we have to train somebody else in to accomplish that same task. And then the risk of failure here. So just like phenotypic selection, genomic selection doesn't always work. And so if it doesn't work, it could cost a lot of money in terms of genotyping, and you may not have made any progress. So in terms of helping to solve these problems, I'll just skip through this. We've put together a collaboration of different public soybean breeders from the upper Midwest, ranging from North Dakota to Minnesota to Iowa, Missouri, Kansas, Nebraska, Illinois, Indiana, Ohio State, and Michigan. And the objective of what we call our SoyGen group, which stands for Science Optimized Yield Gains Across Environments, is to elevate these regional collaborative field trials so they're not only still an important objective of these trials, but so that they're even more than this, so that they're not only used for making variety advancement decisions, but also being used as developing a data resource for doing genome-wide association studies to look at the genetic architecture of these traits and develop genomic prediction models. So how do we turn those into a resource? How do we develop the data sets and the databases to make that data best available? And then how do we ultimately leverage and use all this information to apply genomic selection in everyday public breeding programs? So I've already talked a little bit about the Northern Uniform Soybean tests. So I don't need to mention much more about this, but basically there are these regional tests that we as public breeders use to critically evaluate the yield performance of our potential new varieties. And so, as I said, there's been historical data accumulated on these things for a number of years. And so we wanted to get that historical data into something more usable and analyzable and queryable than in the past. In the past, they've largely existed as either hard copies or PDFs, which are created from Excel files. And what we did is we essentially took a lot of these different Excel files in which these data existed in different formats, and we standardized them. and we've placed them all in a database. And so this is an example here where we have a yield table here for a particular a particular performance test from 2017. All these breeding lines were tested, in this case, at 4 locations, which is on the smaller side for these tests. And we've taken all these different non-standard formats and basically converted them all into a standard format using a series of barcodes. And we've taken all these now and placed them into a standardized format that is more easily analyzable. And so now that's been done on the phenotypic data collection side. And so we've compiled data now going back to 1993 to 2022, and actually 2023 has been done now as well. And we're contemplating going back even further in time, but we haven't done that yet. But within the data set that we currently have, we have 199 different locations represented. We have over 1,600 different environments represented across these parts of the upper Midwest. And as I mentioned before, we've been genotyping everything in the regional trials that we can get our hands on. And so we went back in time as far as we could and got all the available seed that we had that we could get our hands on. And then we've been genotyping everything entering these regional trials since about 2016. And so up until this current time, we have over 3,800 different breeding lines in these trials genotyped. And we have used a combination of the soybean 6K Infinium SNP array. But more recently, we've moved to whole genome skim sequencing performed by Genomic, just because the cost was the same as the 6K. We get a lot more information from the skim sequencing imputation than we could get from the 6,000 markers before. With the Genomic platform, we get like many millions of SNPs off of these lines. So we've analyzed these data and we find a lot of expected things, I guess. We find that largely the germplasm kind of clusters by maturity group because it's easier to make crosses between soybean lines that are from a similar maturity group. We found that actually among the different public programs, there is not a lot of clustering. And so I think this is a hallmark. This is sort of a signature of the regional trials. Regional trials help facilitate lots of germplasm sharing, and the genetic data sort of bears that out, where the structure here among these different breeding programs is actually quite flat relative to land races and private XPDP lines. So you can see there's not a lot of differentiation among the different public programs, although the different programs on this phylogenetic tree here analyzed through the tree mix software kind of are ordered by likelihood of germplasm exchange. I know Minnesota and South Dakota in the past have exchanged a lot of germplasm, and Ontario's exchanged a lot of germplasm with us, and Michigan, Iowa, and so forth. Indiana, Kansas, and Missouri are more likely to exchange germplasm. And nevertheless, the FST coefficients among these programs is actually quite small, and there's not a lot of divergence among the programs, I think, because of the germplasm exchange that these trial system has allowed or help facilitate. So we've done some things like looking at what are the regions of adaptation, regions of the genome that contribute to adaptation to different zones. So if you look north to south, you can see that a major driver in the differentiation between north and south programs is at E2, which is a maturity locus. It also has some differentiation at E3, another maturity locus, and determinancy locus known as DT2. We didn't see a lot of divergence from east to west. I hear stories about there being divergence in soybean germplasm from east to west, but it didn't really bear out in our data too much. Our data didn't show this in the public germplasm. And then we found regions of the genome that have lower genetic diversity in certain maturity groups than others. So you can see at this one region here, you can see that in the early germplasm, there's very little genetic variation, whereas in the later germplasm, there's more genetic variation. So those could be regions that we could target for introducing genetic variation by just sharing germplasm between divergent maturity groups adapted to different parts of the upper Midwest. So real briefly here, in terms of database development and standardized workflows, we're trying to create these things, and so that we can more easily use the genotype and phenotype data we're collecting in the regional trials for implementing genomic prediction, as well as helping other soybean breeders who are collecting genotype and phenotype data within their own internal programs for doing genomic prediction on sort of on an everyday routine basis. And so the data I talked about has been deposited in a relational database now available on Soybase. So thanks a lot to Rex and Jackie for helping with this. You can go in and pull down all these regional trial data in a very organized way, subset, query, do whatever you want. Just pull it all down in a standardized format so you can perform your own analyses. You don't have to wade through lots of different Excel files that have all been standardized and formatted differently. And we also been working a lot with Heather, Amanda, and Keo on developing the soybean base through the breeding insight group and the breeding OnRamp group as well. So all of our genotype data is being deposited in the soybean base. We're using this for our own everyday internal data inside of our own program, as well as depositing the data from the regional trials here as well, the genotype data from the regional trials here as well. And so that's been really, really nice because you can go in, everything is nicely organized. We can have our different genotyping assays here. And with each genotyping assay, we have all the metadata in terms of the maps used and the assay design and sample collection information. All that kind of stuff is stored in the metadata of these different assays. You can pull down these different assays. You can pull down projects within assays, and then within projects, you can pull down accessions that you want to look at. And you can pull it all down in a very standardized format, and they can fit very easily into a standardized data analysis workflow for genomic prediction. So that has been a huge help. And speaking of pipelines for genomic prediction, we are trying to accomplish this and streamline this within our own program so we can reduce the effort it takes to get genomic predictions and ultimately reduce the time to selection decisions, and also make it easier for personnel transitioning between projects to accomplish these tasks. And so this work has been done by a postdoc in my group, Vishnu Ramas Subramanian, who I think is online today, so he can answer any specific questions about this. But basically, he's put together a pipeline of QC, download QC, data filtering, data imputation, model development, training population optimization, and prediction using a combination of either multi-trait models or multi-environment models in a very streamlined fashion using standardized data input, again, made possible through Soybean base. So here are the details on each of those. I don't think I'll spend a lot of time on these details. Be very happy to connect with you offline about these types of things. But basically, once again, data import. And here in the imputation part here, we've been able to execute or implement different types of marker imputation. One particularly powerful version of marker imputation is known as alpha plant impute, from I guess kind of the alpha family of software that's available out there. And this can basically take pedigree information to enhance the power of the imputation. So it's a very powerful imputation engine, and we're making this very readily accessible through this pipeline. So once again, we have multi-trait models, multi-environment models in this pipeline. And then finally, we've placed all the parts of this pipeline into an RShiny app. And so Vishnu has developed this RShiny app. You can go into this RShiny app, upload your phenotypic data in a standardized format, upload the genotypic data from Soybean base, set your target population, set your training population, select trait you want to predict by following this menu. This menu here along the top. Okay, this is the data input table. You can do quality control in terms of filtering genotype data, however you would like to. Again, you can impute using a variety of different methods, including the LDKNNI method. Also, the alpha plant impute method is also available in this RShiny app. And you can set various parameters around that. You can optimize your training population within this application. You can perform cross-validation to assess your model accuracy. And you can choose from a variety of different types of models. So you can choose different genomic selection models. You can, if you want to do a multi-trait genomic prediction model, that will be available. And then importantly, we're also making it possible to do multi-environment genomic prediction models as well. So, and then at the end, if it all works correctly, which I believe it does most of the time, you will get the prediction for your trait of interest for all of your different breeding lines according to whatever model and set of settings you set. And then you get, with each of the predictions, we also provide a reliability of the prediction to indicate to you whether or not you had good connectivity between your training population and your target population. And this app has been implemented in the various different ways. It's available on GitHub, on Vishnu's GitHub page. He's also put this within a Docker, and we are putting it on Cyverse. And so you can access the application through Cyverse and take advantage of their computing resources. So I think I have a little bit of time left here. Sort of in a nutshell here, there's lots of ways to use genomic prediction, and we're actively trying to create the data resources and working with the OnRamp team to create databases and streamlined workflows to make it all possible for everyday public variety development programs. So. And with that, I'd like to acknowledge our Soygen group, the group of PIs here, Brian, George, Matt, David, Nicholas, Martin, Leah, Carrie, Rex, Katie, Andrew, Bill Shapog, Grover Shannon, Danny, Detchen, and Eliana. These are basically our Soygen PIs. We'll all try to work together to make genomic selection more accessible in the public sector, public soybean breeding sector. And then I'd like to again acknowledge my co-authors, Vishnu, Clayton, Rex, Amanda, Heather, Keo, Sushan, Ben, Liana, and all the other new public soybean breeders out there who I've had the opportunity to work with. This work has largely been funded by the North Central Soybean Research Program, the Minnesota Soybean Program, and this is just a picture of our collaboration. So thank you very much for your time and attention, and if there's any time for questions, I'd be very happy to answer them.